So indeed, I represent two institutions, the Faculty of Medicine of the University of Geneva and the Qatar Biomedical Research Institute, where recently I took a position of a scientific director of one of the 3 research centers in it. And it's my great pleasure to speak together with these great gurus of G proteins, Kirill and Henrik. And you know that what we work on in the lab is the pipeline or the workflow of the basic to translational research, which starts from the patient, brings us to analyze the molecular and cellular dysfunctions of the mutant variants, which permits us to establish the high-throughput screening platform, which we use to try to identify drugs which we can reposition, which we then validate in the animal models which we create, and then ultimately the goal is to bring it back to the patient. Speaking of these investigations we do at the cellular and molecular level, we study the things which Kirill has mentioned and Henrik will be also mentioning. So we look at the GTP uptake and GTP hydrolysis. We look at the intracellular localization of the variants. We look at how they interact or couple with RGS proteins, GPCRs, and beta-gamma subunits. And we look at the neomorphic features of pathogenic variants where they gain interactions with the chaperones RICATE-A and RICATE-B. And I'm happy that since we're in Europe, a large part of my team could join me, so you will hear a talk about the dominant GPCR coupling of some variants from Gonzalo Solis. Later today, there is a poster from Denis Solachov on the effects of pathogenic variants on the neurite outgrowth. There is a talk from Ionica Larazati from my lab, who will be presenting another potential repositioning drug candidate, which our screening has discovered. And then there will be a talk of Mikhail Savitsky on the Drosophila models of the disease. And this pipeline, starting with the patient and then bringing back to the patient, implies that we as the academic lab, we must collaborate with the clinicians. And I am really enjoying these collaborations. So just 2 illustrations of the collaboration we have with the team of Giovanna Zorzi here. We recently described 2 unusual mutations in GNAO1, which leads to unusual phenotypes. And then there is another collaboration which we enjoyed with the team from China and the team from France, where we described 2 mutations in the position cysteine 225 to tyrosine or arginine in the French versus Chinese patient, and this gives to quite severe disease. And then, of course, as another recent highlight, we have published this year in print the collaborative paper with Moritz Thiel and Anna Coy on the first-in-human zinc trial. Moritz will be talking obviously about the clinical trial which he is running. But I just want to mention that I was at the Pediatric Movement Disorder Conference in Boston like 2 weeks ago, and the story, I was very happy to hear that it was one of the research highlights presented at this conference. But what I want to focus on today is a paper which is not yet published. Hopefully it will be published like any day. I'm checking my emails regularly now. On unusual variants, which some of you may have heard about. Some of the patients, of course, in this audience know about it. And this is another collaboration, very fruitful, from among us as the academic lab and 3 clinical teams from Spain, Italy, and France, where we have discovered these novel mutations which lead to the entire loss of the switch 3 region in G-alpha-O. So this is the exon intron structure of G-alpha-O. So it contains 8 exons, and then the difference between the isoforms A and B, as presented earlier today, is that basically after the exon 6, all the exons are common between the 2 isoforms. There is alternative splicing. Either exon 7A and 8A are joined with the exon 6, leading to the isoform A, or the 2 alternative exons are taken. And this leads to basically very similar G-alpha-O proteins, which differ by 24 amino acids only. So there are these 3 patients in Spain, Italy, and France, perhaps there are more, which have the mutations in the intron 6. These mutations are shown here, so at the position +1 after the exon 6 or +2. Both, as you can see, affect this GT or GU in the pre-mRNA site, which is key for the splicing as the donor site. So you can appreciate that these mutations, they do disrupt the normal splicing. And the 3 patients, which I'm showing with the permission of the families, it's part of the paper, which hopefully will be published. They have, well, the patients are of different ages, but you see that the prominent manifestation is the motor dysfunction. There is no epilepsy seen so far. Edema and the motor dysfunction is the prominent features, and it can be quite severe. So what we have done, we have collected the blood from 2 of these patients out of 3 with the 2 individual mutations here. And we have done the analysis of what the transcripts in these patients are. And what we find is that there are basically 4 transcripts, which are aberrant ones, which can be identified in both patients with both mutations. This transcript, and then there are additional transcripts which can be seen in one patient, but not the other. Out of these 4 common transcripts, we see those ones where there are different defects in exon skipping and early termination and so on. But basically, the most interesting one I will be focusing on is this one, where we now produce the isoform B as the major protein. And this aberrant splicing producing the isoform B leads to the deletion of 24 nucleotides, which leads to the in-frame deletion of 8 amino acids. So, and this deletion falls on this switch 3 region. So what is the switch 3? The switch 2 was also mentioned earlier today. So if we look linearly, this is the G-alpha O structure with the 2 domains, the RAS-like domain and the helical domain. And then you see these important regulatory sequences or motifs which are spread throughout the protein. And on the 3D model, I show you the switch 2 and the switch 3. This is the model in the GDP-bound state, so inactive state in the complex with the beta-gamma, shown here. So the switch 1 to 3 regions are those motifs in the G-alpha proteins which undergo the most significant conformational changes upon the exchange of the GDP to GTP and then hydrolysis of GTP back to GDP. So specifically for the switch 3, what happens when in the wild-type G-alpha O, when the exchange happens, switch 3 and switch 2 become closer to each other, and switch 3 helps to stabilize switch 2, whose function is actually to stabilize the gamma phosphate of the GTP, and the glutamine 205, which is key for the hydrolysis, is also localized in the switch 2. And I repeat, the switch 3 is there to stabilize the switch 2. And here, this mutant variant removes neatly the switch 3 essentially completely. What I'm also showing you here are some other pathogenic variants which are localized in the switch 3 region, but these are single nucleotide amino acid substitutions or another splice site mutations which leads to the insertion of 2 amino acids in frame in the same region. So looking at these aberrant transcripts which we find in the patients, we find some which would lead to production of highly truncated variants. So these truncations are shown here. So imagine this would be the entire G-alpha O, and one of the variants would basically truncate half of it, and the same for the other one. In contrast, the other variant leads to the full-scale protein production, just lacking this switch 3 region. So for us, not only we want to understand what exactly gets wrong in the patients, this also provides a very interesting pathogenic angle to analyze the role of this highly conserved sequence in the G-alpha O and other G-alpha subunits. because so far there has been no patient described where the whole switch 3 region would be completely removed. There have been experimental studies on removal of switch 3 in the transducing protein, G-alpha-T, but this provides us the pathogenic angle, clinical angle to the importance of this region. So we completed the whole cycle of our molecular and cellular analysis with these variants. So we cloned the delta switch 3 protein. We expressed it in bacteria and also in mammalian cells such as HEC and N2A cells. By the way, we also did the same for the truncation variants, and you see that their expression is quite poor, but the delta switch 3 is expressed at good levels. Then we do the analysis of the GTP uptake and hydrolysis. So you know that there is the cycle, GDP gets exchanged with GTP, GTP gets hydrolyzed back to GDP, which we can monitor in vitro with the purified proteins using fluorescent GTP analogs, such as body-pi GTP gamma S, which is non-hydrolyzable, and body-pi GTP, which is hydrolyzable. So for the body-pi GTP gamma S, there is this uptake of the nucleotide, which we can monitor, and it is the fluorescence stays high because it is not hydrolyzed. For the body-pi GTP, there is an uptake followed by the decrease because it is hydrolyzed. And we can calculate the kinetic constants of binding and hydrolysis based on these findings, on these measurements. And what we find is that the delta switch 3 is dramatically faster in the GTP uptake. There is the K binding is increased about 15-fold and dramatically slow in the GTP hydrolysis. You can see that it basically stays unhydrolyzed with the about 50-fold drop in the K hydrolysis constant. We monitored how these pathogenic variants would respond to zinc, because you know why, and we found that they do respond. This pathogenic variant does respond in the sense that both in the GTP uptake and in the GTP hydrolysis, we see a concentration-dependent drop in the ultimate level of the GTP levels taken, which classify this delta switch 3 in the group 3 of the pathogenic G-alpha-O variants, where zinc affects the basically holding of the GTP. We looked at the interaction with the partners. So RGS-19 interaction is severely disrupted in the delta switch 3, which is actually something which we see for almost every pathogenic G-alpha-O variant. They universally lose the interaction with RGS proteins with very few exceptions. At the same time, the interaction with the beta-gamma is almost unaffected. Well, if we compare the G-alpha B isoform with the G-alpha-A isoform, G-alpha-B seems to interact stronger with the beta-gamma. We don't quite know why. Actually, just a few words. To my understanding, this comparison of G-alpha-O-A and B isoforms, side by side biochemically has not been done before. Maybe I'm mistaken myself. But here we do compare them. They are quite similar, but there are some differences which we find. And the delta switch 3 mutation basically brings this G-alpha-B beta-gamma binding back to the G-alpha-O-A levels. We also looked at the GPCR coupling, and for the dopamine receptor, we basically do not see any difference in this bread assay, but for the muscarinic receptor, we see that the delta switch 3 actually interacts with the GPCR more strongly than the wild-type G-alpha-O or G-alpha-OB do, which may indicate that there is a dominant engagement interaction with the GPCR, referring back to what Kirill and maybe Hendrik will be talking about, the dominant binding, which can affect the interactions of the GPCRs with other G-alpha subunits. And then looking at the intracellular localization of the delta switch 3 variant, we find that as G-alpha-O wild type A or B isoform, the delta switch 3 interacts normally. We see the two locations, the plasma membrane and the Golgi apparatus. We don't see any obvious differences. And actually, empirically, what we have observed is that the majority of G-alpha-O pathogenic variants which maintain the plasma membrane localization, they do not have the epileptic manifestations in the patients with a couple of exceptions. And in this regard, the fact that delta switch 3 localizes normally to the plasma membrane agrees well with this empiric rule that unless there is a massive loss or decrease of the plasma membrane localization, there will be no epileptic seizures in the patient. So we discovered that delta switch 3 is a very strong neomorphic variant. So we looked at the interactions of delta switch 3 with RIC8A and RIC8B. Just to give you a slight reminder, so RIC8A and B, they were initially discovered as activators of G-alpha subunits, meaning they biochemically speed up the GTP uptake by the G-alpha subunit, especially by the nascent G-alpha subunit, which is born like free of any nucleotide, and RIC8s are there to help G-alpha subunit to incorporate its first GTP and then go into the adult life. The RIC8A and B have further been shown to act as chaperones for G-alpha subunits to help them fold after they are synthesized. And actually, if you decrease RIC8A or B levels, this will lead to the reduction in G-alpha O client protein levels in the cells. So RIC8A is known to physiologically be the chaperone for most G-alpha subunits, so there are 16 of them, as you remember, and 14 of them are clients of RIC8A for the wild type, in the wild type state. And RIC8B seems to be dedicated to the G-alpha S subtype, so G-alpha S and G-alpha OLF, or L, as it is called also. And what we have discovered and published last year, that the pathogenic G-alpha O variants, they massively interact with RIC8A, and what is even more exciting, with RIC8B, which is a foreign chaperone for G-alpha O wild type. G-alpha wild type doesn't interact to significant levels with RIC8B. So, and this is what we call the neomorphic activity, because by the definition of the Nobel Prize winner Muller, who studied genetics and laid the backgrounds for the modern genetics, a neomorphic mutation, basically he characterized 5 different classes of mutations: loss of function, gain of function, partial loss of function, and so the neomorphic is another one. So the neomorphic mutation is the mutation which leads to appearance of a new function or a new interaction which does not exist for the wild type allele, or does not exist to the same levels for the wild type allele. In this regard, the interaction of pathogenic variants of G-alpha O with RIC8A, and especially RIC8B, falls under this classification. This is why we claim that pathogenic G-alpha O variants, at least the severe ones, they are neomorphic in their nature. So in addition to this massive binding and trapping of RICs by pathogenic G-alpha O, what they also do is they relocalize RIC8A and RIC8B from the cytoplasm to the Golgi. So look at the RIC8A localization in the presence of wild-type G-alpha O, A isoform or B isoform. RIC8A and RIC8B are in the cytoplasm. But when the cells express the delta switch 3 variant, RIC8A is relocalized massively to Golgi, and the same applies to RIC8B. What we also found is that actually the best meter or the best correlation we have ever found between any biochemical or cellular activities and the clinical severity of the disease is the gain of this interaction with RIC8B. So now, from this analysis, we conclude that, well, we already knew from massive biochemical and cellular activities by many researchers in the past that switch 3 is a key player stabilizing the active conformation of G-alpha O and other G-alpha subunits. What we have done here is that we have characterized the pathogenic variant, a newly discovered pathogenic variant, which leads to a severe movement disorder and developmental delay in patients, and we have characterized this exact variant. So this also allows us to compare the removal of the switch 3 in these 3 patients with the point mutations in this domain. So here, this is a table, which is by far not complete. I created it like yesterday, where we compare some of the pathogenic variants fallen into this switch 3 region. So this one is the intronic mutation, which leads to the in-frame insertion of PQ amino acids in this position here. Q233P is another point mutation, E237K, L, I cannot read what it is, something P, and then finally the delta switch 3. So the point which I want to make here is that here I provide the data on the age of onset of the disease, the presence or absence of the epileptic phenotypes. Actually, all of these patients lack epileptic phenotypes. The movement disorders, all of them have the movement disorders. All of them, or majority, have the developmental delay. All of those tested do not have any MRI abnormalities. And basically, so the age of onset, if we take the age of onset as the meter of the clinical severity, we see that from here to this delta switch 3, we have an increase in the severity. So the onset becomes earlier and earlier. So the delta switch 3 is the most clinically severe out of these other mutations, which kind of makes sense because, you know, it's one thing when you substitute one amino acid by another, but it's another thing when you remove the whole site. If we look at, if we try to correlate this clinical severity, like grading with the biochemical and cellular features, we find that there is a correlation. So looking at the GTP uptake reaction, GTP hydrolysis reaction, and the resulting kinetic constants, we see that this insertion of PQ is actually the mildest. So there is only twofold increase in the rate of uptake and the twofold decrease in the GTP hydrolysis. These two single amino acid substitutions are more severe, but the delta switch 3 is the most severe out of those. And the same applies to the neomorphic activity. Here I show you for the illustration the extent of relocalization of RIC8A or RIC8B from the cytoplasm to the Golgi. For this variant, for example, you still see some cytoplasmic localization. Here you see less, but still some. But for the delta switch 3, this relocalization is more pronounced, and the same applies for the RIC8B. Here you see a lot of cytoplasmic staining left. Here you see less, and for the delta switch 3, you have almost complete relocalization. And we have done the molecular dynamic simulations to try to understand why the removal of this of the switch 3 region leads to these manifestations. And basically, the conclusion is that in agreement with the previous studies, the removal of the switch 3 leads to the relocation of the switch 2 region. Now here the G protein is inverted, so don't get confused. But this is the switch 2 region, and this would be the GTP. And you see, it will be more pronounced after the 200 nanoseconds, that the switch 2 region in the delta switch 3 mutant actually moves far away from the GTP site. And this is quantified in this analysis. So we see an increase in the distance between the glutamine 205, which is key for the hydrolysis, and the GTP gamma phosphate, and the same for the whole switch 2 region swiping away from the GTP. So as a summary to this work, we have characterized novel intronic variants which lead to severe early onset movement disorder, NEDIM type phenotype. These intron 6 splice donor site mutations, they lead to production of a group of aberrant transcripts. Out of these aberrant transcripts, what appears to be the major one is the transcript which leads to the production of the isoform B, so a switch in the isoform selection with the deleted switch 3 region. Delta switch 3 is a severe neomorphic variant with strongly affected nucleotide binding. Importantly, this variant is responsive biochemically to zinc. Delta switch 3 is deficient in stabilization of switch 2 and ultimate adoption of the active conformation by G alpha O. So this provides pathological insights into the molecular role of this important region, conserved region in G-alpha-O. And our next steps together with the patients is that we want to generate IPS, patient IPS-based neuronal cultures to actually measure how much of this mutant protein is present in the cells, because this is something we still don't know. So this was the illustration of, I did something wrong. Can you, sorry about that. We'll have to jump through everything. So I wanted to conclude my presentation by reminding you that we do this workflow of clinical and molecular and cellular and drug discovery based research, which ultimately aims at developing something which is useful for the patients. And here, this is my acknowledgement slide. So I want to, of course, acknowledge the members of my team who are doing this work. I want to acknowledge the clinical collaborations without which this work would not be possible. So you see the names, and many of those names, actually you can see them as faces here. They're present here, and I'm really enjoying and thankful for these collaborations. But I also want to thank the patient associations and patients themselves. So what I have been gradually discovering is that patients actually can be instructive or instrumental in the research we do. This is something which is unusual and unexpected for me, because I used to think, well, we're well trained, we know what to do, and the patients are laymen, they're not biologists, they are not clinicians. So for them it's hard to really influence what exactly we do in terms of details of our analysis. But with more and more examples, what I am learning is that actually patients can be really instrumental in how we bend or redirect our research. There have been several cases in my team investigations when a given package of investigation would not be done if not insisted by the patients. In some cases, I would say, okay, we can do it if you really want it. I don't believe it would work. But suddenly it gives unusual, unexpected discoveries, and this moves the research forward. So I think, at least for me, I'm learning how to engage with the patients, not just as those for whom we try to serve, but those who actually are active players in this research. So thanks a lot.